Okay, so this is where we ended our lecture last time. No? So now we understand that macrophages also play a role in clearing the immune system uh, to the adaptive. No, so let's focus on the M2 macrophages. So we mentioned that the M2 macrophages, they will be reprogrammed, no, from the M1 macrophages. So from the M1, an M1 macrophage can shift, no, into a M2 macrophage, uh, but given certain conditions. No? So the M2 macrophage, the main role of this is to break down the tissues, no, especially the damaged tissues, and it will also take part in tissue repair or what we call wound healing. No? So, the in the aspect of tissue breakdown, no, the main components of the macrophage that will be responsible for tissue breakdown would be the enzymes. No? So here we see in this diagram the enzymes that are released by the macrophages that play a role, no, in removing the damaged tissue. So when there's damaged tissue, your for example, your connective tissue, mostly these are composed of your collagen, which is produced by your fibroblast, and your elastin. So these enzymes that are present in the macrophage, they will release these to uh, catalyze the collagen and the elastin that is found in connective tissue. No? So these are the collagenase and the elastase. No? We also have the plasminogen active. And then we have the role of tissue remodeling. So um, are there people in the waiting room? Kita. Okay naman, di ba? Wala namang naghihintay sa waiting room. Okay. So, when it comes to the role of the M2 macrophages in tissue remodeling, this at this point, no, um, this is the cleanup stage as well. No? So, once they have or the repair stage. No? So once the macrophage was able to clean up, the damaged tissue, it will also clean up the dead cells, the neutrophils, for example. It will proceed to tissue remodeling, or it will also stimulate tissue remodeling. So how does this how does the M2 macrophage help in tissue remodeling? So it will release these fibroblast growth factors to stimulate the fibroblast cells to produce more collagen. And then we have the release of the fibronectin. And second most important is the angiogenic factors. No? So the angiogenic factors, from the word itself, no, angio meaning the blood vessels, and to generate, no, so it's to generate blood supply. The important one here for angiogenesis is the VEGF, or what we call the vascular endothelial growth factor. No? So the M2 macrophage, no, will release the VEGF along with other components. No? We have the protease inhibitor, TGF beta, and any to regulate the extracellular matrix. No? So this will all take part in repairing the tissues, first cleaning up and then repairing the tissues. Can you excuse me for a second? Okay. So this part is clear, no? So the M2 macrophage, we already emphasized, no, the role of the M2 macrophage, no. So this is how it contributes to the anti-inflammatory response of the body. Okay. Are there any questions with regards to the M2 macrophage? Okay naman yan, di ba? Now, the macrophage, no, they also have the function of removing dying neutrophils, no, or dying cells, no. In particular, we are looking at the example of how the macrophage would be able to detect dying cells, no. So, how does this occur? No? So, this is through the interaction of a cellular marker called CD31. So, both the neutrophil and the macrophage contains this marker, the CD31, and in normal function of the macrophage, it will try to be in touch with neutrophils, no? So parang si macrophage, it performs a neutrophil check, no? So lalapit siya sa isang macrophage, and both of them, macrophage and the neutrophil, will contain CD31, no? So the CD31, they will interact with each other, the CD31 of macrophage and healthy neutrophil. And it's like a response, no? It's a response that the healthy neutrophil gives to the macrophage in order for the macrophage to know that this is a healthy neutrophil, no? So once the CD31 of the neutrophil interacts with the CD31 of macrophage, then the macrophage knows that this is a healthy neutrophil and I'm going to leave it alone. So, aalis na siya. Now, when it comes to the dying neutrophils, no, so we mentioned in class when we talked about neutrophils that neutrophils have a short lifespan. No, they will survive for about a week or so, and the dying neutrophil usually will lack the cellular marker, which is CD31. So this lack of CD31 indicates that the neutrophil, is going into apoptosis, no? And it's a very, very early signal that the neutrophil is going to die. Now, once the macrophage comes into contact with a dying neutrophil, the CD31 of the macrophage will not be able to meet, or it will not receive a signal, a returning signal from the healthy neutrophil to say that it's dying na, no? Or to say that it's healthy, no? So, di ba pag healthy, the CD31 will interact. But in the dying neutrophil, there is a failure to reply from the dying neutrophil. So wala siyang CD31 sa cell surface, no? So what happens is that this will result in a reaction, no, wherein the macrophage will determine that it is dying, and it will phagocytose, no? It will ingest and destroy the dying neutrophil. So this is just an example of how this macrophage is able to detect and play a role in the cleanup, no, in removing damaged tissues, no, dying cells. So this is the mechanism by which this macrophage can perform this task. Okay. So is this clear? Walang question dito. Or if you have questions, you can send it through the chat. Okay. And all right, thank you. Sorry, and then you have the chronic inflammation. So we mentioned that the macrophages, they play a role in tissue remodeling. They also play a role in tissue damage, in cleaning up tissue damage. That is mainly by the M2 cells, right? Now, the M2 cells, they also play a role in chronic inflammation. So when you say chronic inflammation, these are persistent infections. It could be persistent infections. Let's say the body is not able to remove a particular antigen or bacteria or parasite, no? So mostly, these types of infections are what we call chronic, no? So, a very good example of this would be a parasite, no, a worm. So a worm would be, even if it is just, let's say, a larva, no, it can be a very large uh, foreign antigen that will take some time for the uh, body to eject, or sometimes no, it's not able to do that. Another example would be the mycobacterium, no, your tuberculosis, diba? When you say tubercles, no, these are lesions, no, parang they are these granulomas that are formed by the body around your mycobacterium, no? Kasi hindi niya mapatay yung mycobacterium. Ang gagawin na lang niya, palilibutan niya yung mycobacterium ng cells so that it can wall it off, no? Hindi, parang it will surround it with the uh, fibroblasts, no, and macrophages, so that it will not spread. So kaya nagkakaroon ng tubercles sa tuberculosis. So these are the granulomas, no. So your M2 macrophage actually is the one that is also responsible for this, no. So those I was giving examples of the chronic chronic infections, no, some examples of antigens that are chronic. And this will result in a prolonged period of tissue irritation. And some macrophages will be destroyed during this process. And some macrophage, no, they will remain in the M2 state. So this state of the macrophage being in M2, um, there are a couple of effects, no? So first, there could be the phagocytosis of the irritant, no, or removal of the damaged tissue. But eventually, no, it will result in the release of cytokines and other factors, no, your interleukin-1, your TGF beta, fibroblast growth factors, and angiogenic factors that will result in the formation of your granuloma. So how does this mechanism occur? So ano ba yung granuloma in the first place? No? So like I mentioned in the example, your granuloma is the walling off of persistent irritants by fibroblast and extracellular matrix. So like in the next slide, I'll show you an example of a granuloma in microscopically, no? And we'll see that it's made up of cells, and the cells are the fibroblast and the extracellular matrix. So yung ano ba yung extracellular matrix ng fibrous connective tissue? So it's mostly collagen, no, that is released by your fibroblast. Kaya tinatawag na fibrosis, no? So these IL-1, TGF-beta, fibroblast growth factors, and angiogenic factors will stimulate, no, the fibroblast and will lead to fibrosis. Fibrosis, this process also cause in other cell types, no, you have the granulocytes, macrophages, and some lymphocytes that will contribute to the granuloma formation. So, yung fibrosis, it is the accumulation of fibrous connective tissue made up of collagenous fibers and fibroblasts. So, this is how your M2 macrophages contribute to chronic inflammation. Okay, so if it cannot phagocytose it, then it will stimulate the walling off of this um, um, of your irritant. Sorry, parang ano, hindi nasama yung slide sa okay. So I wasn't able to include that slide, no. But sige, very quickly I can try to look for a granuloma picture. So this is what a granuloma looks like, no. So your irritant, no, would be at the center of the granuloma. And then it will be surrounded by cells. So mostly these are going to be, so let's look at this one. So at the center of the granuloma, no, para siyang nodule. So here we see, the um, various cell types, no, but mostly this will be made up of your macrophages. You see some endothelial cells here, and then some lymphocytes as well, granulocytes. No, so they will all try to surround this lesion, and it will also stimulate the fibroblast to form connective tissue. Okay, so that's the macrophage and its role in recovery from inflammation. Okay. All right. Yeah, let's go to the next topic, which is cytokines and cell signaling. So for cytokines and cell signaling, there, question. From my understanding, M1 can shift to M2. Can they go back or shift back to M1 macrophages? That's a good question. I think so. Yes, they can. If they survive the M2, so the M2 state. So if, for example, it does not, if they're able to clean up um the tissue damage, and they can be reprogrammed to M2 again. Okay. Sige. That's a good question. Thank you. All right. So let's proceed to cytokines and cell signaling. So yung cytokines and cell signaling, we already introduced this topic in the previous, no. I was thinking we wouldn't include it in this discussion, but masyadong dense yung topic about antigen presentation. So I opted to discuss this instead, no. So this will be the last part of this included in your exam. And there's only a few slides, no? So I tried to summarize this topic kasi um, it's a very complex topic, no? And if you don't have any good background in biology, this is it will be quite a struggle to memorize everything without fully understanding it, no? So let's just try to go back to what the definition is. No? So cytokines is a complex network of molecules that exists and that are responsible for the ultimate control of the immune system, known as cytokines. Yeah. So they are, the name itself, no, comes from cyto, which means cell, and kinin is to move, no. So what it will do is that it will move cells, no. It will send signals, and they will be received by a target cell, and in the target cell we will see an effect, no. So this is very well summarized in this very simple diagram. So um, yung stimulus, if there is a stimulus, in our case, no, the pathogens, no, or the antigens, no, once they are recognized by the PRRs, no, they can stimulate the release of these cytokines, no, and the cytokines will be received by the target cell through what we call a receptor, and this interaction between the cytokine and the receptor will produce a signal that will result in gene activation. And this gene activation, there's so many functions of these different pathways, but the end result is that we're looking for a particular effect, whether it's a normal biological function or um, let's say it's an anti-inflammatory response. So this is how the cytokine works. So we weren't able to mention this grouping in the previous lecture, but it's important to understand that cytokines can alter cell behavior in three different manners. So there's what we call the autocrine, the paracrine, and the endocrine response. So what does this mean? So when you say autocrine, it means that it will be able to act on itself. So we have the release of a cytokine, and that cytokine will loop back and have an effect on the same cell. And then, or from where it was produced. Then you have paracrine. So paracrine means that the signal is being sent to a target cell that is neighboring or adjacent. It is not very far. And this molecule will be received by the receptors that are on the target cell, and then it will have or produce an effect. And then we have the last, which is your endocrine. And endocrine means that there is transport of the molecules, the cytokine molecules, from one particular location to another, and it is distant. So it has to travel, let's say, the blood system or the circulatory system for this cytokine to have an effect. So for exam purposes, the main groupings that you just need to remember would be the interleukin, the interferon, and the TNF family. But this is overly summarized and simplified, as you may understand. There are other families of cytokines, and there are also what we call the chemokines that we have already discussed before. But to look further into these groupings, so the functional grouping of the cytokines, they can be a little bit confusing, mostly because the cytokines have a multitude of functions and effects. However, if we will try to simplify the grouping of the cytokine, we can look at them according to their structural similarities. And mostly, if you understand protein structure, the protein structure of the families is the main difference. So we have the interleukin and the interferon families. They will form an alpha helical bundle, which is constructed mostly from four anti-parallel alpha helices that are arranged in an up-up-down-down configuration. So here we see an example of your interleukin-2. So we see these helices, or helix. And these helices are oriented in a particular manner. So yung orientation niya is this one, up, up, down, down. So somehow, this is common among the interleukin family as well as the interferon family. So sila yung main group ng alpha helical na cytokines. And then we have the group of the TNF, or the tumor necrosis factor. And what we see here is the difference in the configuration of the protein. So it's mostly made up of what we call the beta sheets. So here we see the difference in the TNF. So we have are you trying to sige. Guys, if you have any questions, you feel free to interrupt me. So here we see the configuration of the protein. So we have these sheets. These are called beta sheets. And this makes up this is the main structure of the TNF alpha. So yun yung main difference, okay? And that's generally how we group them, okay? So it's based on the structure. So that's the gist of it, no? But please understand, no, this is overly simplified and for the purpose of immune, no? And also there are other groupings, no? So pwede siyang bifunction, no? But mostly the simplest grouping that you will that you may be able to understand at this stage, no, is the structural similarities between the interleukins and the interferons, and then you have which is the alpha helix, and then you have the TNF, which is made up of beta sheets. Okay, is that clear? Next is the cytokine receptors. No, so the cytokine receptors, they are so it's very complex, no, and how they are become activated, no. However, for simplification purposes as well, no, so let's try to at least remember, no, what are the at least two functional units of a cytokine receptor. So this would be your bale example rin po ng cytokine family, ang chemokines. Yes, pero they are in their own category mostly. I think it's because of the size, no, the chemokines. Pero technically, no, you can also call them cytokines, the chemokines. So I hope that answers. Okay. Okay, so the cytokine receptors, no, they can be divided into at least two functional units, no. So these are the ligand binding unit and the signal transduction unit. Okay, and what we're looking at here on the right side, ito muna, si letter A, is an example of of a cytokine receptor, in particular IL-2 receptor, no. So I chose this example kasi it's quite medyo simple siya. But okay, so there are three main components, no. So you have the IL-2 receptor alpha, IL-2 receptor beta, and the gamma, no. So we have these three pieces right here, no. And the ligand binding unit of the IL-2R is the alpha and the beta, no? So pwedeng ito siya, no? So ito yon, the alpha and the beta. So this is the one that binds to the interleukin 2, no? So IL-2 kasi IL-2 receptor siya eh. And then you have the signal transduction unit. So the signal transduction unit is the beta and the gamma part of this receptor, no? So it's this blue and this yellow, no? So the and as an entire unit, no, this will act as the interleukin 2 receptor, and it's a high affinity receptor, no? So it will bind strongly to IL-2. And so, yun yung tinitingnan natin kanina, no, yung effect ng cytokine, no, it will bind to a receptor and it will result in what we call a signal transduction pathway that will have a particular effect on a cell. So, I don't know if relevant, pero is the arrangement contributing to how fast they respond to a pathogen or infection? Mm. The structural arrangement is mostly so that they are activating the correct, or they are binding to the receptor correctly. So yung different structures specific siya to the receptor, right? So yung interaction na yun, dun siya nagpa-play ng role. But that's a good question. Thanks. So this structure will bind to the IL-2, no? So yun yung tinitingnan natin kanina na na interaction, diba? This signal and the cytokine, as the receptor in this cytokine, no, will bind to, and this will result in a signal. No? And the signal is very complicated. No? So here we see the JAK-STAT pathway. So yun yung next discussion natin. But it is very simplified na. No? So I only have 3 slides left. No? Okay. So I only chose one pathway to discuss, no, that will be included in the exam. I know it's very detailed, pero it's important for you to have an introduction to this, no? Kasi the knowledge and depth of immunology is very advanced now, no? So yeah, dito sa atin, we need more people who understand this, okay? That's why we're trying to discuss this in detail. But yeah, I'm trying to simplify it. Okay, so this signal transduction, no, this is a very simplified illustration or diagram of how signal transduction occurs, no. So there's a receptor, no, and then there's a relay of molecules that will occur, and this results in a cellular response, no. So in the case of the cytokines, no, what are the key components and what are the key events that need to happen in order for this signal transduction to occur, no? And what's the effect? So there's the cytokine, there's the receptor, no? And there's a clustering of the receptor chains, no? So magugrupo-grupo sila. And this will result in the activation of some enzymes, no, the kinases, no. So the tyrosine kinases, they also play a role. They play an important role, no. Uh, this activation is facilitated by the receptor, no. So we see that example here. So we have the receptor. No, there are many molecules that are involved, and this kinase, no, the kinase is one of the important molecules, no? And then you have the activation of possibly a secondary messenger. Uh, let's say, ito na lang yung example na gamitin natin. So we have a primary messenger, and then you have another molecule that needs to be activated that will result the generation of transcription factors. No? So the transcription factors are present in the nucleus. No? And these transcription factors will result in activation of certain target genes that will result in protein synthesis. No? So that's why we have, the 3 most important transduction pathways that are included in your textbook, no, in the topics, because the immune system, no, it will trigger the activation of these 3 very important transduction pathways, no. And to simplify, I will emphasize only 3, and these are the 3 most important, no. It's the NF-kappa B pathway. So the NF-kappa B pathway is the most significant signal transduction pathway for the immune system, no? At least for you to know this, at okay na yon. Sige, yun. NF-kappa B, yun yung pinaka-important na transduction pathway ng immune system. And this is triggered by 2 cytokines, no? The IL-1 and the TNF-alpha. But I think there are others, no? And then you have the NF80, the nuclear factor of activated T cells, and you have the JAK-STAT pathway. So, and the JAK-STAT pathway is a highly conserved pathway of signal transduction. Now, it stands for Janus kinase and signal transducer and activator of transcription. So, those 3 pathways, if you want to try to understand more about these 3 pathways, you can try to look at some references, no. But if you will analyze this diagram, this is the simplified version of the of the NF-kappa B pathway. Ito na yung pinakasimple na nahanap ko, okay? But in summary, no, we have the kinases here. We mentioned the activation of kinase is very important once the signals have been activated, no? And then you have the NF-kappa B, which results in the transcription factors P50 and P65 that will result in the activation of the gene, no? That also influences, no, the inflammatory response, no. So this will generate the inflammatory proteins that will result in inflammation. Okay? And bakit ba yun important, no? So the basis of all our medications, let's say is the molecular knowledge of these molecular pathways, no. So for example, the JAK-STAT pathway. This JAK-STAT pathway, it has an effect on many downstream events, no? So for example, hematopoiesis, immune fitness, tissue repair, inflammation, apoptosis, adipogenesis, no? So with this detailed knowledge of these pathways, no, we can try to manipulate these pathways, no, and find targets that we can alter. For hot dog. Okay. And we can discover, no, drugs, no, that can have an effect on the cell or have a yung have a particular treatment for a condition. So I hope that gives you an appreciation of these pathways, no, and the transduction pathway, or a simple understanding of why these are essential, no. So far, I think that's it. That's the last slide. Do you have any questions? Okay. So if there are none, I will end this, and we will sige, there's somebody in the chat. Hold on. Mag-upload. Yes, I'll be able to upload it now. Yeah, for studying. So I'll provide these and also the quiz, no, so you can answer it during the weekend. Tapos ayan, i-announce ko na lang ulit, no, so we'll have the exam on Wednesday class time. Okay? And you only need to bring pen. Hindi nyo kailangan magdala ng notebook. Yan. Okay? So I'll see you next week.